Hey, KG.
How are you doing, Lance?
Good. How are you?
Good to see you today.
Good to see you today.
Yeah.
So this is our second episode. Last time I was talking to Qiming on the NKG mice.
Yeah, I watched that video. Pretty good one. Very impressive. And learned a lot from the last episode. Regarding the NXG models and the history. And personally, I study oncology back in my postdoc training, and I used personally NSG model a lot. Didn't even know the history of NSG model. I mean, that's great.
Yeah, I heard about it. People have been using this model for so many years without knowing where it came from.
Exactly. I didn't even know there are so many different versions of NSG at that time, because in academia, I think they simply use the NSG, and unlike the industry, they have different choices to pick different version of the NXG models.
Yeah. Okay, now we're on to our topic, which is what the whole genomic DNA humanization model, the next generation humanization models.
Exactly. Yeah, we've been working on this next generation humanized model for
3 years.
4 years.
4 years.
Yeah.
You've been in the company for how long?
Exactly,
for 3 years.
Okay.
3 years. Can you introduce yourself a little bit?
Yeah, absolutely. My name is Kugeng. You can call me KG, and which is the initial of my first name. And I have a background of both molecular biologists and bioinformaticians back in my PhD training at McGill. Studying
McGill. This is we went to the same university.
Same university.
But I'm way earlier than you.
Not that early. A few years.
I was in electrical engineering and computer sciences. Yeah, you're in the medical school,
exactly,
right?
Yeah. Studying. We call it the program is called experimental medicine, and doing basic research basically. And I was studying cardiovascular disease, or specifically vascular disease at that time.
Yeah.
Focusing on the role of microRNA regulation on vascular diseases. And then after that, I went to Toronto, the University of Toronto, studying oncology, changing my subject of study at the Princess Margaret Cancer Center, which is one of the biggest cancer center in North America. And before joining Cyagen, that was a little bit around 3 years ago.
Yes.
And it's been an honor to work very closely with you, Lance, and Cyagen.
Thank you.
And been learning a lot about the animal models that I did not know before, and also having a chance to work very closely with the top university, and as well as the pharmaceutical companies, biotech, biopharmers all over the world. This has been a great years.
Good. It seems that you're enjoying it.
Oh, absolutely.
Okay. Good. Yeah. So today we're talking about this new breed of humanized mouse models. We call it HUGO, Human Genomic Ortholog.
Yes.
Yeah. So somebody came up with this name, which I thought. So what set it apart is that we humanize the mouse gene with human counterpart on the genomic level, as opposed to usually what we in animal model business do, which is to replace the mouse gene with a human CDS sequence of that gene.
Yes.
Right. And so if you allow me to go back a little bit, I can explain to you why we came up with this idea.
Oh, absolutely. I'll be very, very interested.
Yeah. So when you talk about humanization of the mouse, you can humanize mouse on different levels, right?
Yeah.
The reason you want to humanize the mouse gene is that you want to study human conditions, human diseases, how genes are working in human, right? But you cannot do experiment in human. So you put a human gene in the mouse and see how it works in vivo, right? So for example, if you are a doctor and you're a clinician and you run into a rare genetic disease running a family, and through lots of sequencing, you're suspecting certain mutation on a certain gene that is causing this disease running in this family, right? And what do? One option is that you come to a company like Cyagen. Hey Cyagen, can you put this mutation into the mouse gene?
Yes.
Right? We were asked to do it quite often over the years. I noticed it because at one time I was asking our team. I said, "Hey, why those mutation models are all from these doctors, right? And turn out that most likely it's the case that some mutation that is causing human disease. They want to test it out in mouse, but so many times it didn't work.
It failed.
Yeah,
That's the mouse genes.
Yeah, you put a human suspected disease-causing mutation into mouse, the mouse gene, the mouse counterpart. It's a mouse counterpart, but it's not the same. It's got a different context, right? So you put a mutation there, it's not working. I would say, I don't know, close to 50% of the time it's not working. Mm-hmm. So it got me to think that this kind of the simplest version of humanization is not working, a lot of times. So we need to solve this problem.
The transgene got inserted into the genome, whether it's going to disrupt some certain endogenous genes or not. And you're not sure about the copy numbers, and therefore the different generation of the model may be not stable because different mouse may have different copy numbers. And and usually it takes several rounds of passing to get a stable transgenic line, right?
Yeah.
And also another thing is the promoter that I was talking about. It's not driven by the endogenous promoter.
Yeah.
And that's why I think we came up with this next generation humanized model.
That's right. If you allow me to continue, another way of humanization is that you knock out the mouse gene, and at the same time you put in place the human CDS sequence of that gene.
Exactly. Yeah.
Right. But this model, what we call HUGO-GT. GT stands for gene therapy, right? In the area of gene therapy, sometimes the humanized CDS will not work, because sometimes people would like to target not just the protein, but the genomic sequence. Exactly. And sometimes it's not even the protein coding sequence, right? Could be intron, could be UTR.
Exactly.
Yeah. This is one of the incentive that got us thinking that we need a better, humanized model. We need to humanize the mouse gene on a higher level, so to speak. Yeah. Now, if you look at the different ways of humanization, point mutation is one, or you can humanize a section of the mouse gene and put whatever mutation there, right? That's sometimes people do that, right? And also, JAX has a lot of back transgenes. So basically, you take out a segment of human genomic sequence and you just randomly put it in the mouse genome. You don't even know where it ends up with. But it's got the whole genomic sequence, right? It's if you're lucky, expressing the human gene, sometimes, normally. Or appear to be normal. But this is through random integration.
Exactly.
And sometimes you have multiple copies. Mm-hmm. It's not a very repeatable strategy to go with.
And it's not driven by the endogenous promoter either.
That's right. So it's not regulated by the endogenous.
That's the problem of transgenic model.
Yeah.
One thing is the random insertion. You don't know, you have uncertainty there. Where the exactly. We given that, I think right now, I think in today's gene therapy world, people are developing different types of novel therapeutics using different types of technology. So ASO, antisense oligonucleotides, one of them.
Mm-hmm.
People can use it to target either the exons or the introns, or sometimes the UTR, in different methods, or on different purposes. So one is using ASO to skip an exon when the exon is not supposed to be there, or in the opposite case, to include an exon when the exon is skipped by splicing error.
Yeah.
And also ASO can be also used to knock down a gene when it binds to either the UTR or the coding sequence, using another technology. And so I think the big advantages of humanizing the whole genomic sequence, including both the exons and introns and the UTRs. We don't normally humanize the 5' UTR in the promoters because as much as possible, we would like to keep the endogenous promoter driving the genes. Unless, in some cases, especially when people can develop therapeutics to target the promoter, for example, the RNAi machinery therapeutics to knock down the gene by targeting the promoter. In those cases, we also make humanized models with a humanized promoter as well. And talking about the advantages of our fully humanized models, this kind of a one model fits all. Kind of humanized models, and unlike the traditional CDS humanized model, it's good for people who wants to develop antibody drugs or small molecule drugs, to target the protein, or maybe it's good for people who wants to use AAV-based gene therapy, gene replacement therapy, to replace back the healthy copy of the genes back in it. But when it comes to novel therapeutics who wants to target the introns, the UTR, or in some cases the promoter, it's just no longer good for them.
Well, different places along the genomic sequence of the gene, and sometimes you're even targeting the RNA, right?
Exactly. Yeah.
The mRNA.
Yeah, the mRNA.
Yeah. So you want to humanize the mouse on a higher level, so to sum it up, we initiated this HUGO-GT program about 3 years ago. We decided to humanize 100 genes that are frequently studied. Now we have more than that, right? Yeah, so we started out with the list of 100 genes, and so we humanized them on a genomic level, which is replace the gene with a human counterpart. So what we would like to see is that there's no major phenotypical changes. If that's the case, we call it a good wild-type humanization, right? And then you can introduce whatever disease-causing mutations you want to study, right? And although you humanize the gene, the wild-type humanization is supposed to be identical to the mouse, but sometimes you have subtle differences. In that case, you use the wild-type humanization as a reference to the ones with the mutation, right?
Exactly.
Yeah. So there you talked about sometimes you want to humanize the whole thing, including the both UTRs and introns exons, and sometimes just one end of UTR, right? And sometimes you might want to humanize the promoter even, right? So out of this 100 genes we humanized already, it looks like the vast majority of them are working, but sometimes it's not working as we expected.
Yes, I would say 80-90% of the times wild-type humanization would make good, functional, healthy human protein, and in rare cases, I think the human genes expression level is not normal. It's probably the mouse mutation. The endogenous, I would say, mouse promoter may not work well with the human genes, or in some cases, even if the human gene expression level is normal, it may have an abnormal interaction, protein-protein interaction with the endogenous mouse protein, resulting in an abnormal phenotype. So, but I would say majority of them, it worked. The concept is pretty solid.
Yeah. So we got lucky that most of them are working. And even though some of them are not working, we kind of redesign the strategy and try to find a way to get around it.
Yeah,
right?
Exactly. So, but there's a challenge there in rare cases, especially when the gene is too big.
Okay.
Yeah, there might be other small genes, within the intron of that big gene. So
that's right,
if you want to humanize the whole thing, you may interrupt the other genes.
That's right.
So,
that's right.
In that rare cases, we do have our proprietary technology to get around it.
Yeah, that's right.
To kind of still insert the whole genomic sequence at the endogenous site.
So to make these HUGO models, we are routinely dealing with genes, on the genomic level up to 100 kb, even more.
Yeah, even more.
Yeah, so we're talking about quite complicated targeting strategies. So that's why we use our proprietary, improved version of ES targeting, what we call TurboKnockout.
Okay.
Yeah. So a dozen years ago, CRISPR became so popular, and people, started to use CRISPR for everything.
Yeah.
And it kind of forgot about ES targeting.
But not us. We still keep
working on the ES targeting platform. Now we have improved it, in so many fronts, to the extent that we can finish the project, sometimes within 5-6 months. So not too much different if you use comparing with the CRISPR. The upside is that we can do much complicated targeting with TurboKnockout with ES targeting. For CRISPR, if you're talking about gene replacement, knock-in, I would say the upper limit is, in our hand, is about 20K. If it is more than 20K, you still can do it, but the efficiency is so low that you spend tons of money trying forever until you hit the success one. But with ES targeting, we can knock in up to 300-400 kb in one round of targeting.
Wow.
Yeah. So we're about the only commercial company that is providing such kind of projects. That's why we're using the ES targeting, the TurboKnockout, for all of the HUGO projects, right? Yeah. So among these 100 targets, I guess they're focused on which disease areas?
Yeah, because I think in gene therapy, we try to target, be a bit more targeted in disease areas. So there are, I would say, 3 major disease areas we'd like to target. The first one is ophthalmic diseases, and the second one is neurodegenerative or neuromuscular or muscular disease. I put it in the same category, the same assets. And the third one is metabolic diseases. And so for these diseases, they're pretty much a good fit for those novel gene therapy technologies. Either with that being said, either it's ASO or CRISPR gene editing, or siRNA knockdown. And so I think we can cover a little bit more about the different models that we have developed in today's interview. Let's get into some examples. Which gene you want to talk about first? Well, let's cover let's talk about the ophthalmic disease.
Ophthalmic disease, the eye disease.
Yeah, because that is actually the platform we were developing when I first joined Cyagen.
Okay.
We developed this eye disease CRO platform together with all those eye disease models.
Yeah.
And the reason why we focus on eye disease is because it's actually a good disease area that fits the gene therapy world. The reason behind this is that I think there are the multiple folds. The first is that because of the presence of the blood-retinal barrier, and especially when you want to deliver a novel product, a novel therapeutics that toxicity or safety profile may be a concern, and due to the local injection in the eyes and the presence of the blood-retinal barrier, almost no product will enter the circulation.
Yes.
Because they cannot pass the barrier.
So it's safer.
It's safer. It's a safer profile.
Yeah.
So, and a lot of these ophthalmic diseases are caused by a single gene mutation. It's Mendelian disease.
That's right.
Call it, right.
That's right.
So it's a very good fit for gene therapy, regardless of whether you're using AAV-based gene replacement therapy to bring it back, to bring the healthy copy of the gene back, or you're using a gene editing technology to fix the mutation, or using ASO to kind of do either exome skipping or exome inclusion, or targeting one single gene.
So that's why so many companies are working in this area.
Exactly, using gene therapy is the pretty good fit. And also, because we are doing local injections, right, and the material it's so not big, and when it comes to AAV-based gene therapy, for example, which accounts for about, I would say, 60% of the gene therapy vector used in the market nowadays.
Yes.
And production and manufacturing of AAV is so expensive, especially when it comes to systemic administration.
Yeah.
That's why the FDA-approved AAV-based gene therapy is so expensive today.
But the dosage for the eye could be
much smaller, and lowering, significantly the manufacturing cost.
That's right. That's right.
Therefore, the cost of the drug.
Yeah.
And that's why it's I would say a perfect fit for gene therapy. And So we have a focus on retina diseases, and I would like to cover a few ophthalmic disease model we have made in the past few years.
Sure.
Each one have I'll try to give examples that are more representative in terms of using what types of technology to treat the disease. So the first one I would like to talk about is the ABCA4, model.
Yeah.
So the mutation in the introns of ABCA4 will cause abnormal splicing events, resulting in abnormal skipping of exon 39, or sometimes both 39 and 40 in the gene. And because ABCA4 it's such a big gene, right?
Yes.
It cannot be simply, although it's a loss of function mutation, it cannot be simply cured by AAV-based gene replacement therapy.
It's too big to be packaged.
Too big. Its coding sequence is around 7 kb. It can be packaged by, right now today they have the dual AAV technology.
But the efficiency is
exactly.
lower.
Efficiency is not good, and I don't think they have good clinical data, if any. Right? It's all preclinical. It's not a mature technology today. So that's why people right now, in I would say most cases, developing either I would say CRISPR technology to do gene editing to fix the mutation.
Yes.
And ASO to do exome inclusion, bring back the exome 39 that is mistakenly spliced out by the splicing error. One important fact is why we're doing the HUGO model on ABCA4 is because the most prevalent mutation is in the intron. If you want to work on it, the
CDS model wouldn't work.
Yeah, it wouldn't work, right? So you're not actually studying the mechanism underlying. So this is a good example, a perfect example of why we want to make the HUGO model an advantage of making this fully humanized HUGO model.
Yeah, that's right.
And another example I would like to talk about today is the USHR2A model.
USHR2A, that's a big gene as well.
That's a big gene as well, Usher syndrome.
Yes.
And it's even a bigger gene. The coding sequence is about 15.6 kb.
Yeah.
It cannot be delivered by even a dual AAV.
And the genomic sequence is even
bigger.
Gigantic.
Exactly,
it's like 800K or something, if I remember correctly.
If you want to do a fully humanized model, it takes like 3 rounds of ES targeting.
3 rounds of ES targeting.
To make a fully humanized model. And funny thing is that the most prevalent mutation of USHR2A, it's a mutation in exon 13. It's a 2299 del G mutation. It's deletion of one single nucleotide at position 2299. And that single point deletion would result in a frame shift mutation and a premature stop codon, resulting in non-functional Usher protein.
Yeah.
And so the fun thing about the technology, the same ASO technology to cure USHR2A is kind of the opposite of ABCA4. ABCA4 is to bring back exome 39, and for USHR2A we use ASO to skip exome 13 because
to bring the shift back to normal.
Exactly. Bring
frame.
Bring it back to in frame.
Yes.
Because of the deletion, we want to just simply take out the whole exomes. Although it's missing an exome, but the protein is still functional. It doesn't have a premature stop codon.
Yes.
So that's another model we have developed pretty successfully now.
Okay. So in this case, do you humanize the whole gene?
Yeah, we have different models in USHR2A, and the first model we generated, it's a partial humanized model for now because the most prevalent mutation is in exome 13, the 2299 del G. So a lot of people actually are focusing on this particular mutations. So we actually humanize not only exome 13, but some region around it, so that people can work around it. So you can target the intron, flanking the exomes or some nearby sequence as well.
Do you also care about the promoter sometimes?
I don't think so in this particular model.
Not in this model.
But not in this model. But in some models, yes. For example, the RHO model. So the rhodopsin model. So rhodopsin is actually one of the most prevalent, the mutation in rhodopsin or rhodopsin causing retina diseases are actually one of the most prevalent RP
in RP diseases.
In RP disease, retinitis pigmentosa.
That's right.
In the clinic, and definitely the most prevalent autosomal dominance, that's gain of function.
This is gain of function.
Exactly.
disease.
Yeah, and in the clinic, and so the reason why it's not a good fit for AAV-based gene replacement therapy is because it's a gain of function mutation.
That's right. That's right.
So it's not about you lost the function of this protein and you bring it back.
Okay.
Now you have to work around the misfolded.
You might want to get rid of the misfolded protein.
Exactly. So one of the most prevalent mutation in IHO, it's P23H mutation.
That's right.
This particular point mutation caused misfolding of the rhodopsin protein and resulted in the unfolded protein response machinery triggering apoptosis and cell death. So when you look at the OCT image or the pathology, you can see a very clear cell death in the outer nuclear layers where the photoreceptor cells are located. The photoreceptor cell is a lot thinner compared to the normal mice. And so in this case, there are different ways to treat that. A regular gene replacement therapy is definitely not an option.
No.
Yeah. So you can either use gene editing technology to fix the mutation, or you can design ASO or siRNA or RNAi machinery to knock down the genes.
Knock down the faulty gene.
So that's why in this particular model, since people are designing different types of siRNA or ASO that can target different area of the gene. Sometimes the 3' UTR, sometimes the coding sequence, sometimes even the 5' UTR or promoter.
Even promoter.
Exactly. So in this case, besides generating a normal HUGO models, we also generate another version of the HUGO model with even the promoter humanized. And but we don't normally do that because we as much as possible, as I think I mentioned earlier, we try to use the endogenous mouse promoter to drive the human genes.
Because you want to keep the
normal expression as much as possible.
Because you don't know if the human promoter would be working, would be compatible with the mouse
transcription factor.
That's right.
Yeah, exactly. And so in this case, that's why, because we know that someone is developing, siRNA to target promoters. So we make this promoter humanized models as well. And
in this case, it worked.
Yeah, exactly.
Okay.
So we didn't know that before, but luckily it worked. And but I think I heard before we are also developing an AI tool to predict transcription factor binding.
Yeah, that's right. We do have a pretty strong AI team, as you know. We have developed quite a few AI tools. One which is right now widely used and cited in over 50 papers is our splicing prediction tool. So we can predict if there's a mutation, if it's going to cause the splicing problem. And as we speak, we are also working on another AI tool, which I think is going to be very useful, which is to predict if, let's say, if we humanize a promoter, if it's going to work, if the promoter is human, is it going to work in a mouse environment?
Nice.
Yeah. So we're still working on that. There are, you are bioinformatician yourself, right?
Yeah.
And there are a lot of the traditional bioinformatics models, but they're not that accurate.
Yeah. That's exactly what I used back in my PhD training. That was like about 10 years ago already. So when I was still bioinformatician, using different tools to predict microRNA binding size as well as transcription factor binding size to study protein-protein interaction, gene-gene interactions. And because the traditional bioinformatic tools for transcription factor binding size, simply based on the result of wet lab, IP experiments, looking at the binding motif of different transcription factors.
Yes.
But
there's so many motifs and so many transcription factors.
And some motifs are so short, like 4 to 5 nucleotides. This is basically all around the sequence. So when I did the prediction, it was like there are a certain motif I'll try to find whether this particular transcription factor can target this particular gene, but we found out that this transcription factor can basically target every single gene. So because the motif is so short, it's everywhere.
This is the thing, that the biology is so complicated, right?
Exactly, yeah.
And sometimes it's just impossible to follow the logic to come up with some, conclusion, right? Yeah. That's why AI can come in handy.
Exactly, yeah.
And you treat it as a black box, and you just train the model by brute force. If you have enough data, you train the model enough, it's gonna, figure it out by itself. Yeah, we don't even know how.
Yeah, I think it's like perfect timing with the emergence of this large language model.
That's right.
But that was like when 3 years ago.
3 years ago, yeah. 3 years
ago.
The
first ChatGPT announced.
I would say 3 years ago.
Yeah, somewhere around that. And it's amazing.
It was. Right? Well, I remember. OpenAI came up with this thing, right? But even before that, a few months before that, Google, there was a news leaked by a Google engineer, who talked on social media saying, hey, he was so scared. He was in the team developing the large language model, right? And he was talking to chat. Yeah, he was like, hey, this AI is talking to me like he's
a real person.
He's like a real person, and we're bringing this thing alive. And it's so scary, and it looks like the guy was fired by Google, right? That's so funny. And then OpenAI came up with this thing that shocked everybody, right?
Exactly. Yeah, this is what we were thinking. I think maybe we can use AI. Treat AI as a person who understands the language of biology that we don't understand.
That's right.
Find the logic behind, or find the pattern that we cannot see.
Well, you know what? This thing is just not capable of doing everything. So we have our limit, basically.
Exactly.
Yeah, biology is too much for us. Too much for this. Yeah. So we need to resort to the AI, better machine.
Exactly. Better machine.
Yeah.
Going back to our topic, I think, we were talking about the ophthalmic disease models we have developed, and I'd like to talk about one more, which is the TCF4 models. In this case, the first 3 models are all retinal disease models, and this one is actually affecting the cornea, the front part of the eye. And in the gene of TCF4, there are certain number of repeats, CTG repeats, in intron 1, and in rare cases when the repeat exceeds a certain number, it's causing problem.
Yeah.
Because the repeat will cause a secondary structure of the RNA, and the secondary of the RNA will affect RNA binding protein interactions, as well as expression level of the genes. So, and in this case, it's causing a problem in the cornea. So in this case, there are people, I know that someone's trying to use CRISPR technology to cut out the whole intron that contains the repeats, right? So, and in this case, it's also important to use our HUGO models. Because if you only have the CDS, you won't even have the phenotype. To be honest, right? Because it doesn't have the repeat expansion.
That's right.
And so basically, I used 4 different examples to cover different way to treat genetic diseases in terms of eye disease.
Yeah.
The ASO for exome skipping, or ASO for exome inclusion, or ASO siRNA for knockdown, targeting sometimes the UTR or promoter.
Yeah.
And also gene editing technology.
Yes, that's right.
To cut the repeats in the intron.
Yeah.
So so actually, whatever technology you're using, I think
the HUGO model will allow you.
We fit it. Yeah, exactly.
That's right.
Yeah.
What about other disease areas like metabolic diseases?
Yeah, and for metabolic diseases, and so I can give 2 examples. The first one is the PCSK9.
Oh, this is a hot one.
It's a very hot one, exactly. So the PCSK9, the protein normally interact with LDL receptors.
That's right.
Leading to the degradation of the receptor, so that normally the function of LDL receptor is to bind to LDL cholesterol and clearing the LDL, the bad cholesterol,
cholesterol,
in the circulation. So when there's a certain mutation in PCSK9 or upregulation of the protein, it causes excessive degradation of the LDL receptor, causing a higher LDL cholesterol in the circulation. So in this case, people can develop siRNA treatment to knock down PCSK9. And so there's an approved drug right now on the market using siRNA to target to the 3' UTR of PCSK9. And so that's the reason why we don't only humanize the CDS, but also including the 3' UTR in our humanized model as well.
So have the HUGO PCSK9.
Exactly. And another example is the ATTR model.
ATTR.
So TTR is a transthyretin protein. Either the abnormal metabolism of the TTR protein or a certain inherited mutation of the protein will cause abnormal metabolism or deposition.
Yes.
And because this is misfolded, so it causes deposition of the protein in the heart. Normally the protein should be in the bloodstream, right, in circulation, but the misfolded protein depositing in the heart will cause stiffness of the heart and making the heart more difficult, challenging to pump the blood out of the heart and causing cardiac myopathy eventually. And it's a very severe problem, and people can also use siRNA to target, to knock down the TTR protein.
So you target the 3' UTR?
Yeah, sometimes the 3' UTR, sometimes the coding sequence. And I understand that sometimes people can even target the 5' UTR as well. But I think, so regardless, we don't know which sequence or which region our potential clients may target. So we develop a model that fits all, humanizing the whole thing. So
and this is a quite popular model.
Oh yeah, exactly. Yeah, and whichever area you want to target, our model would fit your needs because it has everything there.
That's right.
And yeah, because this is the regular CDS humanized model or even regular mouse model cannot fit because they're particularly designing a siRNA sequence to target the human sequence, and in some cases not only the coding region. So there are many examples as well, but I'll say in terms of metabolic diseases, and in many cases, or in most cases, the models we develop good for siRNA-based knockdown of the mutant or misregulated genes. And speaking of, I think, unlike the last part, it's the neurodegenerative.
Neurodegenerative disease.
And the neurodegenerative or neuromuscular or muscular would put them in the same category,
like DMD.
Same as. And I can also give a few examples of the models that we have developed as well. The first one is DMD.
Yeah, that's a gigantic gene.
It's the biggest genes in human, with what 79 exomes, spanning 2.5 million.
2.5 meg.
Exactly.
Well, that's why I was challenging our team to humanize the whole thing, and they're still working on it.
That would take so many rounds of US targeting, right?
Well, you'll never know. Maybe one day they'll come to me with the good news.
But we have successfully made some partially humanized DMD models already.
Right? Some of them are already, like 500-600K. In length.
So the challenge about DMD models is that not only does it have 2.5 million base pair genomic sequence, but it doesn't really have a hotspot mutation, right?
That's right. It's all over the place.
People found thousands of different types of mutation.
That's right.
In that gene, because when you have that big of a gene, it's actually prone to mutations. Because our regular DNA, replication machinery.
That's right. It's just
prone to
by
error.
statistics, because it's so
Exactly.
big, it's prone to error.
Exactly.
Yeah.
And since there's no hotspot mutations, it's challenging to kind of develop a one product fits all therapies.
That's right.
But there is one people developing the mini dystrophin or micro dystrophin by AAV.
That's right.
Gene replacement therapy.
There are a couple of drugs approved already, right? And more in clinical stage.
Yeah, but I don't think the clinical data is promising. It's very promising.
Well, marginal so far.
Yeah, I would say so. So that's why people are actually many people are actually developing ASO therapy.
ASO-based therapy
to do exome skipping. And Sarepta, I think, got several approved therapy targeting different exomes.
That's right.
And exactly the models that we have developed. So we have developed a model with exome 44 and 45 humanization.
Yes.
And with exome 44 deleted on top of it. So that deletion of exome 44 will cause a frame shift mutation and premature stop codon. And when you use ASO to skip exome 45,
so both
of them now
you bring it back
into
frame.
Exactly.
Okay.
Although it's missing 2 exomes, but
still
it's still a functional protein. It's still better than the micro or mini dystrophin that with only
Is that right?
Selected numbers of exome and domain.
Okay.
And since there are 79 different exomes in it and different domains, you really have to. That's why there are so many different versions of mini micro dystrophin in the market, because you really have to do different types of combination to find the best combination that have that has a good functional protein and fits within the capacity of AAV to deliver, which is 4.7.
That's right. Yeah.
And so that's why people actually developing different types of ASO technology to do exome skipping. So the one targeting exome 45 is one of them, and there are also ones to target exome 51 or 53, I don't remember. Yeah, 51. This is approved drug by from Sarepta. And the good thing about the one targeting exome 51 is that it's good for someone who has deletion on exome 50 or exome 52.
Oh.
So or exome or from both from exome 46 to 50. So different types of patients. Because when you skip exome 51, so it bring it back to frame for all those patients actually. So although it's not considered as a hotspot mutation, but at least one therapy can fit multiple types of a patient with different types of mutations.
Wow.
And so the reason why people are still developing different types of ASO drug in DMD is because, like I said, there are thousands of them.
That's right.
So it's hard to cover every single patient.
And dystrophin is a structural protein, right?
Yeah.
So missing a couple of exons wouldn't make it too bad. It's most likely still working to like 70-80%.
Yeah. And so just like the ophthalmic disease that I cover in neuromuscular or muscular diseases, there are also different. I would say we have made. I'm going to use 2 different examples to cover one that is good for exome skipping technology for therapy, and the second one I'm about to talk about is the SMA.
SMA, that's a pretty successful model we developed.
I would say this is the very first human model we developed,
model.
Right?
That's right.
The very first HUGO model that we have developed, even before I joined Cyagen, or at the time I joined Cyagen, we're developing. I don't remember.
Yeah, that's the very first one.
The very first one.
Yeah.
And we have pretty decent data.
Oh, very good data. If I remember correctly, we used all the approved drugs. The Zolgensma, right? The HUGO model. 2 years ago, I was in Ionis. We gave a lunch and learn there, and we were introducing the data, and I was like, it looks a little bit awkward for us to present this model to you guys, since you have developed the drug, the ASO drug, a few years ago. But these guys, they look at the data, look at our strategy, for the HUGO, they agree that this is a better model. They would have used it back then when they developed the Spinraza. Yeah. So basically, it's just replace the mouse SMN gene. Mouse has only one version, right? Yes. With the human SMN2, which is the mutant version. Yeah. And so the model without treatment would die.
In a few days.
And
within 10 days, I remember. That's it.
Yeah, if you treat them. Oh, I remember the small molecule risdiplam. Yeah. If you treat the mouse with any one of the three drugs, right? Zolgensma, Spinraza, risdiplam, the animals are gonna live forever. This is so dramatic. I'm not a scientist, but I look at the results. It's unbelievable. Yeah.
And just an amazing model that compared to the existing model, which is a bit, I would say, artificial.
That's right.
Because in human we have two
copies.
Yeah. Right? SMN1, which is the normal one.
Yeah.
So SMN2, which has a mutation in exon 7, causing a splicing error, excluding exon 7.
That's right.
And so when you have the mutations in SMN2, it excludes exon 7 in around 90% of the transcript. So it maintains only about 10% of the regular functions. Then you have, let's say, if you have a loss of functional mutation in SMN1, so if you have only one copy. The good thing is that different people have different copy numbers of the SMN2. So if you have only one copy, you have only 10% of the function. So if you have several copies, so it's that's why you probably have only very mild, or you don't even have any phenotype. And so in our mouse model, we replace the mouse normal SMN genes to the human SMN2, which has the mutation in exon 7. So at the endogenous side, that's why it's driven by the endogenous machinery, unlike the pre-existing one, which is the transgenic model with a random insertion and the artificial delta 7 there. And so I think historically people found that persistent model not stable. The reason behind it is probably it's not an endogenous replacement, so the expression level is not normal. And our model has normal expression level at the endogenous sites. And so for this particular model, it's a good fit for ASO-based exome inclusion neurotherapies. Just the opposite of the DMD model I covered. DMD is good for exome skipping, and this one is exome inclusion.
That's right.
Bring back exome 7. And the two neuromuscular muscular models for our HUGO program. And another one is the last one I'm going to talk about. It's neurodegenerative disease models. The Friedreich ataxia. In the FXN genes. So it's also this is the model of repeat expansion in the intron as well. So also the repeat expansion in the intron. Normally in normal people we have only a few repeats, no more than 36 repeats. And when you have more than that, it's causing problem. And in some patients it can go up to hundreds.
Wow.
Or even over 1,000 repeats.
Wow.
Yeah. So so it's in the intron, right? So when you have that many repeats, also misfolding, would of the RNA would cause an issue, right? And so it's also the HUGO model is also a good fit, because if you only do if you only do CDS, you don't have a phenotype.
That's right.
And that's why people in some company they do a transgenic model. So just to insert a copy of the human genes with the hundreds of repeats into random places. So, but this type of model, since it's random insertion, it has some uncertain. It has some level of uncertainty there.
Yeah, I heard a lot of complaints about those kind of transgenic models. Sometimes what you see, you think it's the phenotype, but actually it's the artifacts. Yeah, it's pretty hard to differentiate.
and sometimes it could be unstable because you may see a phenotype in one mouse but not the other.
That's right. Yeah.
So this is another example which is a good fit for HUGO model generation. People can use gene editing technology to kind of cut out the whole intron without affecting, hopefully, the CDS, the protein.
That's right. Yeah. So we covered quite a lot.
Yeah, 3 I would say different disease areas. Ophthalmic diseases, neuromuscular diseases, and metabolic diseases, which are actually the 3 major platforms. We have SSCRO.
Yeah, that's right. So the HUGO concept is kind of gaining traction. And I don't know how about you. Sometimes I got asked, "Hey, we need your HUGO model. We need HUGO this and HUGO that."
Exactly. It's it's drawing a lot of attention, and I would say, scientifically, a lot of people actually approve our concept.
That's right. Yeah. And also we also draw sense of satisfaction being able to help these guys.
Exactly.
You know, it doesn't matter if they're basic researcher or drug developer. They use the HUGO model to develop better drugs. Understand better the mechanism behind the disease.
Better the underlying mechanism and better fit for efficacy.
That's right.
For their therapeutic products.
That's right. So we're developing more of these. So we have a long way to go.
Exactly. Exactly. And we have expanded our pipeline to, I think definitely over 100, probably 200 right now.
Yeah, if you count all the mutations, it's going to be way more than this.
Yeah.
Good. Yeah, I enjoy talking to you guys. I myself is a non-scientist, and I do enjoy talking to scientists.
I would
like
say you are right
now. We have how many? Around 30 PhDs in the company. I'm going to talk to every single
one of them. Yeah. And,
okay.
Thank you, Len.
Yeah, it's a pleasure talking to you, and I'm looking forward to the next episode.
Exactly.
All right. Yeah.